Hello, my name is Sarah Lepin. I am an MD PhD student from McGill University, and it is a pleasure to present you my poster. Mislocalization and aggregation of TDP43 is the pathological hallmark of ALS. Furthermore, mutations in the TARDBP gene, encoding TDP43, have been associated with ALS. However, the consequences of these mutations remain poorly understood. Using CRISPR-Cas9, we engineered two homozygous knock-in iPSC lines carrying mutations in TARDBP, encoding TDP43A382T and TDP43G348C, which are two frequent ALS variants of TDP. We aim to explore the pathogenic properties of these variants in human motor neurons derived from induced pluripotent stem cells. Knock-in iPSCs and the isogenic control were differentiated into motor neurons using a well-validated protocol that mimics normal development. We first confirmed, using qPCR and immunocytochemistry, that the mutations do not impair differentiation of iPSCs into motor neurons. Further immunostainings with two neuronal markers revealed that TDP motor neurons form a normal axonal network with comparable total axonal area and branching. To examine whether those cells were more susceptible to cell death, we quantified levels of cleaved caspase-3, a marker of apoptosis, and found a non-significant trend towards increased levels. We also analyzed the viability of motor neurons over a span of 6 weeks and observed that TDP cultures survived equally well as the control in medium without or with neurotrophic factor supplementation. Postulating that TDP motor neurons may be more vulnerable when challenged with a cellular stressor, such as glutamate, we next compared the viability of cultures after glutamate treatment. We observed only a trend towards reduced viability of TDP motor neurons. Taken together, these results indicate that TDP motor neurons do not display a neurodegenerative phenotype. To assess the functionality of motor neurons, we recorded the spontaneous activity of cultures using multi-electrode array, or MEA. We observed a significant decline in mean firing rate compared with control with prolonged time in culture. Additionally, we noted fewer active electrodes in TDP cultures, indicating that they are more electrophysiologically silent. To further examine the mechanism underlying this, we performed co-immunostainings for pre- and post-synaptic markers and found significant differences in puncta counts and size, indicative of synaptic pathology. Finally, we analyzed protein levels of synapsin and PSD95 using Western blot. Levels of synapsin were significantly decreased at 6 weeks post-plating, coinciding with the observed decline in firing rate. However, SYN1 transcript levels remained unchanged, implying a post-transcriptional mechanism. Overall, our results support that synaptic dysfunction is an early event in ALS pathogenesis, preceding axonal defects and motor neuron loss. Thank you for stopping by my poster.